Okay, hi, we're back. Sorry about that. Hopefully, you guys can all get online again as you were here, but you are gone now. Um, but now I can see what you're saying. So come on in and join us, please. Um, okay. Hi, Aristotle. Okay, so this is a live stream. Sorry, it might bounce a little bit here. This is a live stream to help you review for the Unit 5 exam, covering everything from DNA structure all the way to biotech. Now, keep in mind, if you're not my student, it's possible that the biotech things covered could have been a little different, but by and large, I would imagine a lot of the content is the same. Okay, so while I wait for questions, and feel free to just pop in and ask questions anytime, I'll just do a quick review of the topics that you should know for the exam. I think that's pretty consistent across all different teachers. So you're going to need to know DNA structure. That's going to include DNA and RNA. To know the processes of DNA replication as well as protein synthesis. You also need to know gene expression and let's see what else. Gene expression, biotech, protein folding, mutations. I think that covers everything. My students, unless you're third period, which I haven't given to you yet, there is a review sheet in Google Classroom. Also, again, my students, and I think also Mr. Hajarian for sure, and I think Mr. Gan's students, I believe are, again, I can't, you got to ask your teacher, make sure, but I believe for all of us, your A-level question will be something in the way of a small concept map where you'll be given terms and you need to connect those terms. But again, with Mr. Gan's students, I would confirm that with him. I think that's what he was thinking of doing, but don't take my word for it. Okay, so whatever questions you guys have, go ahead and I guess let me know. Otherwise, I'll just kind of babble about stuff until you guys have questions. Okay, so can I explain translation in protein synthesis? Totally. So remember, the process of translation is how you get from the mRNA, which is like a copy of your one recipe, your protein, right, into a protein. So it all happens on the ribosome. Ribosomes are located in the cytoplasm. They can either be located just free floating in the cytoplasm, or it could be a ribosome that's attached to the rough ER, and that's why they call it the rough ER. The ribosome has two subunits. So think of it almost like a kind of almost looks like a hamburger bun, right? There's a small subunit and a big subunit, and they clamp around the mRNA to read it and translate it. So the actual message of the mRNA, or the message of the protein, like how to decode it, is in the mRNA. And any ribosome can read any mRNA, right? What it's going to do when it first starts translation is it's going to scan that mRNA. It's going to find the start codon. So very important, when you're translating a protein, even though you might have the beginning nucleotides, you actually don't want to start translating from the beginning. What you actually do is you look for your start codon, which is AUG, right? AUG happens to code for the amino acid methionine, right? And so don't worry, those of you who are asking questions, I will totally answer them. I'm just going to do this first. So you have AUG, and once you find AUG, then you start counting by threes, right? Then you start reading in codons. So you find AUG, and then from AUG, then you would read the next three codons and the next three, right? And you're going to use that codon table that I gave you in class. So whatever the mRNA says, you look on that table to tell you what the amino acid that would be, right? And the way, what actually happens on the ribosome is it will start reading the mRNA by having the tRNAs come along. The tRNA has an anticodon. The anticodon is the complement to the codon. And so, Sneha, I'm answering your question right now. So the codon is on the mRNA and the anticodon is on the tRNA, right? So they're opposites, they're complements. So if the mRNA is AAA, then that means on the anticodon it will be UUU, right? But the way that we usually read the mRNAs we read it is by mRNA. So if you see on the mRNA AAA, then you would look on your codon table for AAA. And then with the codon table, right, that's going to tell you which amino acid goes with AAA on the codon. So very important. So codon table is not an anticodon. Yes, Kevin, don't worry. I've got all that. We'll get to all that. So the idea is you've got all these tRNAs flying around, and the tRNAs each carry only one passenger. That one passenger is a specific amino acid. So like the tRNA that brought the methionine for the start codon only carries methionine. And there's another tRNA that only carries leucine. And there's another tRNA that only carries proline, right? So it's like trial and error. The tRNAs fly in, and if they match, the tRNA and the mRNA match, the anticodon and the codon are complements, it stays and it drops off its amino acid. If it doesn't match, it goes away. And so what the ribosome will do is it will take the amino acid off of the tRNA and attach it to a chain of protein or amino acids so that you build your protein, right? And so once the tRNA has dropped off its amino acid, it can go off and find another of the same amino acid. And pretty much translation just continues that way until you get to a stop codon. Now, again, you don't need to know the stop codons by heart, but it's easy to remember as you are awful, you are gross, you go away, or you are great, you are awesome, you go away. So once you get to the stop codon, the idea is there is no tRNA that's going to match it there. So it falls apart at that point. There's actually a protein called a release factor that will bind there, and then the whole thing falls apart and translation's over. Which leads me to the next question that people are asking, which is protein folding, right? You might have a particular sequence of amino acids, but now for the protein to take on its actual function, it has to fold in a particular way. So this is where protein folding comes into play. The idea is that the amino acids will fold in a very specific way. And so you're asking about the rules. The general rules are as follows, right? You have some amino acids that like water and some that don't. The ones that don't like water are called hydrophobic amino acids. The ones that do like water are hydrophilic. The hydrophobic ones, then typically proteins exist in a cell in the cytoplasm, and the cytoplasm is a lot of water. So it would make sense that those hydrophobic amino acids would run away from the water by trying to go inside the very center of the protein, right? If you think of it's not really accurate, but if you think about the protein like an orange, right? You have like the outside of the orange and the middle of the orange, and the middle part of the orange doesn't really get in contact with the outside of the water if you're washing it, right? So hydrophobic amino acids will go on the center, and then the hydrophilic ones are on the outside. Hydrophilic is more general though, because it just means they don't mind the water. So those are going to be your positively charged, your negatively charged. I have a question about my door. The door to my left? Okay, hold on, Kevin. So anyways, we have, I'm at a dance studio. I'm not at home. That's why it's a different door. My daughters are in dance class right now, so I'm live streaming while I wait for them. Hopefully that answers your question. That's why the door looks different. Okay, so amino acid wise, right? Your hydrophilic ones are going to be positively charged and negatively charged, or they could also be ones that have hydrogen bonds. So the general rule of thumb is anything hydrophobic goes on the inside, and then positive and negative attract. Positive and positive would repel each other because positive, like the same charge doesn't want to be near each other. Negative repel. And then the ones that were green, which you don't need to know about the color, but the idea is in the demo, we also had some that could form bonds, and we said those were like green and green. Those were hydrogen bonds. So those are the general rules that I would say, like if I asked you how a protein might fold, you'd want to be able to talk about those types of interactions. And the overall idea is though that when you have a protein and it gets to a specific shape, then it has a specific function, because the shape determines the function of the protein. And that's going to tie into mutations later, right? Because the idea is if you change the order of the amino acids, then you're going to change the shape. If you change the shape, you potentially change the function. Okay, I think I've gotten now, hopefully, the first couple questions from Manya and Aishwarya and Aristotle. Sneha, I also kind of went over the anticodon and codon. Josh was asking about biotechnology. For my students, what I want you to know is what's in those basic notes, right? So I want you to know what the Human Genome Project is. I want you to know what epigenetics is. I want you to know the basics of what genetic testing is, what bioengineering and CRISPR. Again, just the basics, not super complicated stuff. I would also want you for each of those to kind of be able to talk about what are some of the issues that are out there, right? Like, why is it something that people have to discuss? Why is it something that's ethical implications, and why might it be controversial and things like that? Let's see. Let's talk about epigenetics for a second, because I'm seeing a lot of questions about those. So epigenetics is the idea that you have the same DNA in all of the cells of your body, right? Like every single one of your cells has the same instructions. It has the same humongous recipe book of how to make every protein that makes you. And yet, we don't make every protein in every cell, right? So epigenetics is, well, gene expression, let me start there. Gene expression is the idea that only in each cell, only some of the genes are switched on and translated and transcribed and translated. And then others are actually not transcribed and translated, right? And so epigenetics is the study of that. Epigenetics is looking at, first of all, the idea of like, how do we switch on and switch off certain genes so that we are or are not making certain proteins in each cell? And not only that, how does that now translate into heritability? Meaning, if I, in my lifetime, have certain experiences that cause certain genes to be switched on or switched off, how is that potentially now being passed down to my children? They used to think it was, okay, you just, throughout your life, epigenetic tags get put on that switch on genes and switch off genes, and that ultimately would have an impact on that person's health or well-being, right? But now what we're also finding is that some of those changes can actually get passed down to your children. So it could be that some of the things that my genes now, some of the genes in my cells that get shut down may have been passed down to my children, and then they might pass them down to their children, right? So it's possible then that some of the epigenetic changes, it doesn't change anything about the instructions, like none of the bases have changed in my DNA. But what might be changing is the instructions of which ones to switch on and switch off. And so there is evidence to say that even going back several generations, right, that those epigenetic tags are actually getting passed down to your children and your offspring and your future generations. So that's sort of the idea behind epigenetics. It's still a very new study. There's a lot we don't know, but it's sort of the idea of how do we study that? And then also the idea of like, can we switch some of those tags? Like, let's say some certain genes have been switched off in me. Is it possible for certain medicines or molecules to now go and undo that, like to switch something back on? Because then it might actually have a lot of power and ability to be like a medicine, right? Like, what if we could give someone a medicine that would now just switch a gene on that we needed to switch on, or switch a gene off that was accidentally switched on? That's sort of the idea behind epigenetics. Okay. Let me try to address a bunch of the questions. Kevin, I see you have a lot of different ones, so I'll just work my way through that in the context of other people's questions, okay? Joshua, well, actually, let me finish with CRISPR. So CRISPR is a particular type of biotechnology that we talked about. And in this case, CRISPR is gene editing. So it originally came from bacteria. It was a way for bacteria to like find a virus that had infected them in the past and like basically get rid of it so it didn't do it again. But scientists, and the way it worked was it would save a copy of that virus in its like own DNA, so that the next time it saw it, it would basically use another protein called Cas9 to like, and then if we did that, the Cas9 would recognize that sequence, and when it found it in the virus, it would cut it up, right? I'll address the A level in a second, guys. And so what scientists have now done is they've been able to modify CRISPR and Cas9 in such a way where they can tell the bacteria, instead of like, oh, bacteria just randomly look for whatever has infected you, they can now actually program CRISPR to say, this is the sequence I want you to look for. And so now CRISPR has the ability to find that sequence, cut it out, and now potentially change it and edit it and, change it in a person's DNA. So it's, again, very, very new, but it's sort of the forefront of all sorts of things that might now be possible. And again, that brings with it a lot of ethical questions of should we or shouldn't we do those things. But CRISPR essentially is gene editing. Okay. So in terms of gene expression, right, epigenetics, just to sort of finish off gene expression. So epigenetics is essentially the study of how those gene expression might be, inherited down the line, right? Gene expression also involves other molecules. The idea that you have the same instructions, but we have certain molecules that will switch on or switch off other genes. So transcription factors typically bind to your DNA and serve as like a cheerleader to say, hey, RNA polymerase, come here. So it makes transcription happen more. Repressor proteins tend to do the opposite. They bind and they're like a physical roadblock, so they don't let RNA polymerase pass, and then RNA polymerase can't translate. There's also RNA processing, which we talked about as part of gene expression. So the idea that in eukaryotes, but not prokaryotes, we have a middle step where you do transcription and then RNA processing and translation. So RNA processing is where first you add things to the beginning and the end of your RNA. That helps it to get out of the nucleus. It helps it find the ribosome. And then it also kind of protects it in some ways, like from enzymes that might break it up too early. And then what also happens is splicing. That's where you actually look at the RNA sequence and you take certain pieces out and then glue the stuff together. Jennifer, I see you have a question. Don't worry, I'll get to that too. So the idea is that when you have RNA splicing, you cut out the introns. And remember, that's the confusing part because it almost sounds like introns should stay in, but it's the opposite. So introns come out. And then after those introns come out, then you glue all the exons together, and that gives you your final mRNA that goes to be translated. Let's see. So I think I've gotten through biotech and epigenetics. Let me talk about Joshua's question, which is chromatin, chromosomes, and DNA. Yes, they are all related, right? So DNA is the nucleic acid, which is made up of nucleotides, which is double-stranded, right? Double helix, all that stuff. Chromatin and chromosomes are technically two different forms of the DNA. So during most of your cell cycle, when your cells are needing to access the DNA and do protein synthesis off of it and do DNA replication, it is loose and it's uncondensed. So that's what they call the chromatin, which is sort of a very, not tightly wound strand of DNA. But when it comes to something like mitosis and I'm getting ready to divide my DNA and I don't want it to be a big jumbled mess, then it gets condensed. And so essentially what happens is it gets wound around histone proteins to get condensed. And so that would be technically what we call a chromosome. Sometimes people will sort of interchange the idea of chromosome as like a segment of DNA, which is fine. But technically we would refer to, so they're all related because they're both made of DNA. Chromatin is the uncondensed version. The chromosome is the condensed version of the DNA. Jenny, you asked about bonds. I assume you're talking about bonds involved in this unit. So let's talk about that. So you have, within a single strand of DNA, you have covalent bonds that hold the strand together, right? But then when you have the double-stranded, and now you have like the ladder, what holds the two strands of DNA together are hydrogen bonds. So for DNA, those would be the two types of bonds you'd be looking at, right? Covalent bonds to hold the single strand together, hydrogen bonds to hold the double strands together. With proteins, you have now slightly different bonds, right? Because you have hydrogen bonds in the DNA, so you have to break those during transcription. Then you're going to have When you're building your RNA, you're actually constructing covalent bonds between the nucleotides that form the RNA. And then you also have, to build a protein, you have to put peptide bonds between the amino acids. So I don't need you to know any specific proteins. I just want you to know how you make proteins in general. Oh, no worries. Let's see, introns and exons. So again, so you have like a beginning, like a rough draft of your RNA. And remember what you do is so you have beginning piece of mRNA, and then you cut out the introns. So it's like if you're editing an English paper, like you take out a sentence, but then you like make everything else flow together. That's what's left is your exons. Let's see. Let me try and not miss any questions here. I think I got, okay, so let's talk about DNA replication for a second, and I'll keep going down the list. DNA replication is copying DNA, right? It's how you get your sister chromatids. Remember, it happens during interphase, and this is the one where there's a lot of different enzymes involved. So you have helicase that unzips the DNA by breaking hydrogen bonds, and then primer is put on by an enzyme called primase, which I didn't give you the name of the enzyme, so don't worry about that. But the idea is you put on an RNA primer, sort of like a hook to get started, and that gives DNA polymerase 3, which is my main building enzyme, something to hook onto. And so it starts to build the DNA, right? But then at the end, you actually have to cut out the RNA that you put in as your hook, replace it with DNA, so that you can finish off DNA replication. And all of that would happen technically in the nucleus. Mutations are changes in DNA, right? So we talked about all the different kinds of mutations, but the idea is anytime you change the DNA, then you might change the RNA. If you change the RNA, you might change the protein. When you change the protein, you're going to change potentially the shape. So in your notes, you should have all the different kinds of mutations, right? Point mutation, and a point mutation could result in a silent mutation, missense, nonsense. And then remember, frame shift is where everything just shifts over. So like everything's weird and messed up. So study guide wise, remember the study guide is not complete. The study guide is only DNA structure and protein synthesis, etc. Like it's missing for sure, like biotechnology, epigenetics, gene expression. So no, don't just study. You should study everything. Okay, let's talk A-level question. And again, if you're not my student, then check with your teacher. But for my students, your A-level question will be some sort of mini concept map where I'll give you like 7 to 10 terms, and you'll have to put them in a map and connect them appropriately. Jennifer, I think you were asking about the difference between the bonds. I'm not sure what kind of bonds you mean, so I'm just going to take a wild guess. There are hydrogen bonds or covalent bonds. Covalent bonds are way stronger. So peptide bonds, which are covalent bonds, and then the bonds between nucleotides, those are way stronger. But hydrogen bonds are fairly weak, so it's fairly easy to break them. Joshua, yes, positive and negative, they don't retract, they attract, right? So yes, there are positively charged and negatively charged amino acids, and when they're near each other, they want to get close to each other. And then hydrogen bonds are between other hydrophilic amino acids that also help them stay together. Let's see, I don't see any new questions, so feel free to throw anyone on if I missed it. Let's see, what else can we talk about? Talk about protein folding. Talked about mutations. Let's see, you should know the structure of DNA, right? So at the most basic, it's a nucleotide, sugar, phosphate, and nitrogen base. So let me check something. I see something buzzing. Okay. Okay. Let's see. Are there hydrogen bonds between hydrophilic? No. So generally speaking, well, yes and no. Without getting it too complicated, for the sake of the protein folding part, I'm going to say that generally that's not what's contributing to the folding of the protein, right? So you can assume that hydrophilic wants to stay with hydrophilic and hydrophobic wants to stay with hydrophobic, and they kind of just stay away from each other. I mean, it's a little more complicated than that, but for our purposes, that's good enough. Okay, let's do tRNA, mRNA, RNA. Sure. So all of them are produced through transcription, right? I mean, if you think about any kind of RNA has to be made through transcription. And in this case, no, there are hydrogen bonds in protein folding. Sorry, I misled you there. There are hydrogen bonds in protein folding, RNA, mRNA, and tRNA. So there's 3 kinds of RNA, right? mRNA is the copy of the DNA that you make so that you can get it out to the ribosome. The tRNA are the RNAs that are shuttle buses, and they carry each one specific amino acid to the ribosome. So each one only carries one specific amino acid and no other. And then rRNA, which I think for some reason kind of seems to throw people off the most, rRNA is essentially part of a ribosome. So the ribosome is think about your car. It's got a lot of different stuff in there, right? So the ribosome has a lot of different stuff, and you can classify everything in the ribosome essentially into mostly those 2 things, protein or rRNA, right? And so rRNA is the 3rd type of RNA, which is making up the ribosome. And essentially they think it might play some of those roles in like taking off the amino acid and attaching it to the polypeptide chain, things like that. But again, they all are important, right? But they just each do different things in the transcription and translation processes. Anything else you guys can think of? Questions? Let's see. Trying to think of what else I can tell you all. I'm going to scan here and see if you asked any other questions that I missed, because I know there was a period where there was a lot of questions and I didn't quite get them. Oh. Yes, Joshua, I will repeat it one more time. So what I said about hydrogen bonds is, yes, there are hydrogen bonds in protein folding. There are absolutely hydrogen bonds. What I meant was there are not hydrogen bonds between hydrophilic and hydrophobic amino acids. You would only see hydrogen bonds between 2 hydrophilic amino acids. You wouldn't see them between 2 hydrophobic ones, because hydrophobic amino acids, that portion, like the aspect we're talking about in terms of the 3D folding, those would not be able to form hydrogen bonds. So yes, there are hydrogen bonds in protein folding, but they're only between the hydrophilic ones, not between hydrophilic and hydrophobic, or hydrophobic and hydrophobic. Technically, you can kind of think of the hydrophobic as they have essentially what you call hydrophobic interactions. It's the idea like, I don't like water, you don't like water, let's go not like water together. But not hydrogen bonding. Okay, what else? Let's see. So I will post the answer sheet to the review sheet online tomorrow after 3rd period, since my 3rd period hasn't had it yet. I would say, one of the things I would say you want to make sure you can really do well is you want to understand all the processes, of course, of transcription and translation and protein synthesis and DNA replication. But what you don't want to do is mix all the terms up. It's really easy to do. So make sure you're really clear on those. Yeah, so essentially, you don't want to, we're not going to go too much into gene expression, but I want you to kind of think about, in terms of transcription rate, it just means like, how fast am I making my protein, right? So if I have a gene and there is a transcription factor binding to that gene, it's like a sign, it's like advertising that's saying, RNA polymerase, come here, you should make this protein, right? And so it's going to get transcribed more, which means now the rate of transcription goes up. On the other hand, if I have a repressor protein or a methyl group, which is something that was in the epigenetic videos, right? And the idea is that if these molecules now bind to a gene, and instead of making them more attractive and saying, hey, come on and make this protein, they actually do the opposite, they shut it down, then that would decrease the rate of transcription, right? You would be making that protein a lot less, or maybe not at all. Okay, so let me give you a few tips in terms of like how I would remember certain things that I think are really easy to forget. First of all, when we have the names of enzymes, remember they tend to name enzymes things that tell you what they do. Now, not always, but in one specific case, I would say you'll be able to do it easily in that we have RNA polymerase and we have DNA polymerase, right? I mean, the difference is literally one letter. And if you're not careful, you're going to get them confused, right? So remember, the name of the enzyme helps you to know what it makes. So RNA polymerase makes RNA during transcription. DNA polymerase makes DNA during replication. Now, there's technically two different DNA polymerases we talked about, but they're still involved only in DNA replication. So that should be sort of an easy thing to remember, hopefully. The other thing I think people mix a lot up is transcription and translation. So, their names are very similar. You want to make sure you remember the order. So transcription comes first, translation comes second. Transcription is all things RNA. When you're making RNA and you're trying to, make a copy of whatever type of RNA, but it's transcription. So that's going to be done by RNA polymerase. It's a very multi-purpose enzyme, right? It's the one that unzips and puts the nucleotides together. So it's all purpose. Whereas, and that starts at the promoter. So the promoter is the beginning. It's like the start sign that tells RNA polymerase, start here. Okay. And once it starts there, it would build the RNA, which is made of ribonucleotides. So when we talk about nucleotides, it's a much more generic name, right? Which would include the nucleotides in DNA and RNA. Ribonucleotides are more specific. It's referring to the nucleotides that make up RNA. So that's going to have the sugar ribose and the nitrogen base and the phosphate. DNA replication is that much more complicated process now where you have a lot of different players, which I went over earlier, right? Helicase kind of tells you what it does. It unzips the double helix. And then you have primase, which again, I didn't really ask you about, but it puts on the primer. And then DNA polymerase, which is one that makes the polymer of DNA and possibly also cuts out the RNA, right? And that's the DNA polymerase 1. So with translation, right? So transcription starts at the promoter, but translation starts at the start codon and ends at the stop codon. So it's entirely likely you're going to have a piece of RNA where there's a little bit at the beginning that doesn't get translated, and then there's a little bit at the end that doesn't get translated. What else? We just have a couple more minutes, so hopefully we've gotten most of your questions answered. Yes, so the beginning of transcription is you have all your DNA, right? It's just in the nucleus, it's hanging out. And then what's going to happen is the RNA polymerase will come and bind to the promoter, which is like a start sign, most likely because some transcription factors were sticking there and said, hey, come on over here, right? In contrast, a repressor protein would be a protein that sits on a different gene maybe and says, no, don't bind here, right? So a repressor protein is a protein that binds to your RNA or your DNA so that transcription won't happen, whereas transcription factors typically bind to the RNA or the DNA to help encourage transcription. And so once it binds to the promoter, then it would just start transcribing from there, right? And it's going to keep going until it reaches the stop sign, which we didn't talk about what it's called, so don't worry about that. Chromatin, Joshua says, chromatin is wrapped around histones. Yes. So no. DNA is the nucleic acid. It doesn't make DNA. What chromatin is wrapped around the chromosomes to make is DNA is wrapped around the histones to make chromosomes. So we call DNA chromosomes technically when it's in its condensed form. But essentially, other than that, it's all nucleic acid, right? I mean, it's all made of the same stuff. It's just a question. So think of like DNA almost as yarn, right? Where it's like wound around like a ball to make it more compact and tight and easier to move around. That's like a chromosome form. Versus if it's just loose, then it's called chromatin, but it's still made of DNA. A nucleosome is what we call that DNA wound around a histone, like that's the name. Other questions? We'll go a little longer, but I can't go too long. But we'll go a little longer since I know I had some technical difficulty at the beginning. So we'll go like 5 more minutes if people have questions. If not, we'll just cut it off, up to you guys. So we didn't learn about the nucleosome. That part, that's why I said that was the optional one in your concept map for my students. Again, not my students, you should check with your teacher. We did talk about repressors. If you check your gene expression notes, it's one of the things I told you during gene expression, the idea that repressors or repressor proteins are the ones that bind to a gene to sort of make it less likely for transcription to happen. But that's all I need you to know about them. What else? Anything else? I'm trying to think of other things I could tell you all. Well, let me take a look at our review sheet while anyone sees if there are any more questions, and we'll take any questions you might have. Yes, I can explain RNA splicing again. RNA splicing happens. What happened? Okay, so RNA splicing is just the idea that I'm going to edit my RNA before I actually translate it, right? So it means that I'm going to, there are enzymes, you don't need to know about them. Essentially, there are enzymes that will cut out the introns and then glue all the exons back together, and that's RNA splicing. Histones wrap around, DNA wraps around histone proteins, and that's called then the chromosomes. So think of the histone as like a protein ball, and then the DNA is like the yarn that's going to wrap around it. Together, that thing's technically called a nucleosome, right? But then when it's all condensed like that, we say DNA is in the chromosome form instead of the chromatin. RNA splicing occurs every time in eukaryotes, but it never happens in prokaryotes. No, chromatin is basically, think of chromatin as my uncondensed DNA. So it's a double strand of DNA. It's just not wound around anything, right? So if I'm going to give you an analogy, I would say if yarn is my DNA, then chromatin is like my yarn just kind of in a big messy pile on the floor, right? But then my chromosome is when I take that yarn and I put it into like little balls by wrapping it around little golf balls, where the golf balls are the histones, and then the DNA is wound around it to make it much more compact so it's easier to move around. Yes, DNA is wrapped around the histones. But it's not single-stranded. I mean, it's double-stranded, right? I mean, it's normal. It's a double helix. Any lingering questions? Remember, you can also ask me questions later. I mean, I know this is not the most ideal, timing for everyone. So you can email me with any questions. So DNA is further wrapped around. Yes, essentially, the DNA is technically just that nucleic acid, just that biomolecule, right? But then in order to condense it and make it more packaged and easier to transport, especially during mitosis, it gets, condensed essentially by wrapping around histones. I'm not sure what you mean, Manya, in terms of when transcription and translation in prokaryotes. Transcription and translation happen the same way in prokaryotes as it does in eukaryotes. The only difference is, since prokaryotes have no nucleus, everything happens in the cytoplasm. And for prokaryotes, there's no RNA splicing, right? There's no RNA processing. So it just goes transcription straight into translation. Okay. Okay, so we're nearing the end here. If you have any more questions, fire away really quickly, but otherwise, I should probably get going. So chromatin wraps around histones to become chromosomes. Joshua, it's all still DNA, so I wouldn't say it's it doesn't become DNA, it's already DNA, right? The yarn is still yarn, whether it's in a pile or if it's wrapped around a golf ball. But I would call the yarn all by itself, like not wrapped around the golf ball, chromatin. And then I would call the yarn wrapped around the golf balls chromosomes. Okay, any last questions? Unfortunately, I do have to run soon. I have to go pick up my kiddos. But like I said, you can come see me tomorrow, brunch, lunch, tutorial, I guess Friday, right? Or email me with questions. I know in the past you guys have emailed me questions and I will totally answer them. Okay. So have a good evening and let me know what other questions you guys have. And we'll call that a night. Okay, so I will see you all later. Third period, I'll see you tomorrow. Oh, yes, repressors prevent protein synthesis. Okay. Any last ones? You're welcome. All right, have a good night, everyone. Get some sleep. Have a nice dinner. And I'll see you guys later. Okay. Bye-bye.